We are on the other side of a Nobel Prize for AlphaFold. How many AI-discovered drugs have been approved to date?
I think 0, but I do think that should be changing very soon.
Drug approval odds, they have stayed flat at about 8 to 10% straight through the genomics, CRISPR, machine learning revolution.
There's this spring, which over time we've been able to layer in more and more and more technology. And at some point that spring surely has to just snap.
Is the current state of virtual cells an emperor with no clothes?
Humans are just not very good at understanding biology. Math is the right approach to describe physics, and AI will be the right approach to capture biology.
The holy grail of AI drug discovery here is basically to set up this autonomous scientist loop.
I think it's inevitable that eventually AI will just overtake and augment in a large part of the discovery process.
Would you dare to put a year on your estimate when the first AI discovered drug actually gets?
I think there's a high probability that within the next year it will enter the clinic.
My guest today is Rico Meinl, the head of applied AI at Retro Biosciences. Welcome to Axelar Bio.
Thanks for having me.
So we are on the other side of a Nobel Prize for AlphaFold and several multi-billion-dollar rounds by AI-native biotechs, including Retro. So the good question to ask is, how many AI-discovered drugs have been approved to date?
Starting with the easy ones, huh? I think if you're applying the hard filter, I think 0, technically after phase 3. But I do think that should be changing very soon.
So I guess that's a good setup. And I would love for you to walk us through where on the curve of AI drug discovery than we are today.
The obvious thing that sort of changed over the past couple years is we've been extremely good at designing new molecules once a target is known, or being able to design a binder against it, an antibody. Certainly we've made some strides in being able to select targets with novel approaches. How well those approaches work for actually real novel target detection, I think is a bit to be seen, and of course to be validated in the clinic. But I think at this point in 2026, it's safe to say that having a target, even if it's a difficult to target one, being able to inhibit it with a sort of more engineering friendly modality like an antibody, which is not as much of a black box as many small molecules are, has become significantly more accessible, which I think will lead to a huge wave of AI discovered drugs. It's just of course, with the clinic being the major bottleneck, it will take a while for those to trickle in.
Oh man, there is already so much to unpack here. So let me try to digest it. So you're saying basically we are getting pretty good in using different AI models as proven across different companies, hitting different benchmarks on designing a specific protein to hit a specific target. So that's the earliest stage of proving that maybe there is something interesting there. But then you're saying we basically are not really sure yet how well those targets will translate into clinical stages and into patients, right?
Right. Surely for the targets that we had already known before that we're now able to better target, I think there's a higher probability those will translate. For the truly new targets, of course, we'll have to see in the clinic. I think one important point here that is potentially underappreciated, some, a bear case would be that, we've been able to make antibodies against many targets before. There's plenty of CROs that are able to generate antibodies and binders to hit a target. What's actually new here? I do believe that just having the ability to, within individual 96 wall plate screens, to discover a binder against a target that maybe didn't have a strong binder before, kind of one shot the design, and just having a feedback loop that's much faster than spending maybe months to work with a specific CRO, also having to screen the CRO before and then having to work with them to get to the same stage. I think that will unlock a bunch of downstream capabilities. I think biotech is one of my sort of, long-term frustrations is that I think a lot of a lot of horrific stuff happens downstream of slow feedback loops, and any feedback loop that can be accelerated, even if it's sort of like just in the early stages of like drug discovery, is a huge win for the field, and will probably trickle down to like sort of the next bottlenecks.
I want to present both the bull case and then the blockers. So starting with the bull case, what is like the best specific example, like an oh shit moment on some benchmarks that we have hit at the early stages, either within retro or some other AI biotechs that we can point to right now?
I mean, okay, like for me, that was an interesting moment where sort of like to introduce this sort of like maybe like 2-3 years ago, obviously protein structure models have been around for a while. Being able to take an amino acid sequence and determine the structure of a protein, solving the protein folding problem. But then sort of like this next wave was the protein language models, where a lot of the protein folding is essentially enabled by having these like massive sequence databases, where essentially you're able to determine like coevolution of amino acids that are then close together in the structure, which learns a good prior to be able to do structural prediction. But we can also just like train language models on these massive sequence databases. And that enables interesting applications for just like sequence generation or variant prediction. And I guess like for Retro's use case, we were interested in this idea of reprogramming cells to a younger state, like take an aged cell from like your skin, for example, and bring it back to a younger state. Ideally, we can do that, of course, like in vivo, but there are some safety hurdles. So even being able to do this in vitro has interesting clinical applications, because you can take certain cell types, reprogram them to a younger state ex vivo, and then put them back in a safe manner, and give someone like a rejuvenated blood system or rejuvenated immune cells for the brain. So we were interested in this idea of, can we take this reprogramming process, which is basically like taking some old skin cells from a person and dropping these 4 proteins on them, to bring them back to, over like a 30-day process, to this rejuvenated stem cell state. And at this state, you essentially erase all of the accumulated aging marks, except for some, like you don't fix DNA damage mutations, of course. Maybe you select against the ones that are heavily mutated, but you don't, the reprogramming process doesn't actually fix any DNA mutations. I guess nobody would claim that. So the problem is that in this process, it's very, very inefficient. So what we were wondering is, can we just re-engineer those proteins to increase the efficiency of this process? And to not linger on this for too long and sort of give you some space for asking specific questions, we took a language model that we trained together with OpenAI. We regenerated 2 of the 4 proteins that are used for this reprogramming process, and sort of like the wow moment was that our proteins were able to reprogram 50 times more efficiently. And the starting rate is maybe like 0.1% of cells that you start with actually turn into stem cells, and we were able to increase this by 50x.
And so the way you did this is basically, you're saying prior to this, there were these protein folding models, such as AlphaFold is the best example. They were using protein data specifically. And what you did, you applied that, but you combined it with LLMs. So in that instance, specifically OpenAI's model, which is capable of general reasoning, and that's resulted in a much stronger prediction model.
Yeah, that's a great point. I mean, like essentially I guess the utility of AlphaFold for these proteins was not very high, because they tend to be very unstructured. Their structure exists in this sort of like ensemble state, where they would just like have these floppy regions that would only form structures when they interact with other proteins. So when you put these into AlphaFold, you essentially end up with like one structured core. That part actually binds to DNA and regulates like, what sort of gene programs you would activate in the cell, maybe like the reprogramming program or the rejuvenation program. And then the rest of the factor would just be like sloppy and unstructured. And there's like not much information there, and which is also why people have considered these guys undruggable, because there's no pocket for us to stick a small molecule, which is why essentially a protein language model enabled us to just like work in the sequence space. Yeah, so like we essentially took a large language model, GPT-4o at the time, and sort of like added a bunch of native bio data about like protein sequences, protein structures, and then we applied it to this task of re-engineering those proteins. And we had some other use cases that were actually for structured proteins. This is where the structure part came in handy, but for those guys, it was just sequence based.
So just to understand this correctly, I mean, we have had machine learning or AI, whatever you want to call it, in bio for over a decade now. But are we saying that the bull case is we are reaching, or we have reached, a new inflection point now because for the first time we are combining these old style ML models in biology with general reasoning LLMs, and that yields potentially a much more powerful combo?
I think there's like two sort of separate threads of the past couple of years. And I think your framing is somewhat correct that like it's not necessarily that these models haven't been around for, I think like BERT, for example, like the first sort of like mass language model that people would use for, you know, like learning language representations came out in like 2018, if I remember correctly. So these models have certainly been around for a while. I think there's like a bit of I think there's like two things. First of all, like the sequence databases have been like steadily growing, and like I mentioned, the sequence helps you learn like a good prior to be able to do structure prediction, for example. At the same time, I think there's a bit of like a 4-minute mile phenomenon here, where you know, like AlphaFold enabled like a whole wave of machine learning researchers to be like, wow, there's some really interesting stuff in biology. It's like a extremely interesting domain to be working in. What can we actually do with the existing data? And if you look at the protein data bank and what people have been able to do with, like, you know, designing new binders and antibodies, it is largely taking the data that we already had for many years and sort of like restructuring the training data sets to be able to extract like new alpha, to be able to gain new capabilities that we didn't have before. I just like, and just to add to that, and then there's a whole other sort of like space of like large language models being able to just help with all sort of like processes in the drug discovery process, and being able to use these like native bio models to sort of like, you know, autonomize and drive a lot of the drug discovery process for us.
So that's an interesting point. So you're saying right now there are a lot of gains to be made by using the most state-of-the-art AI models or training new types of AI models on existing data sets we have. And it seems like we're making a lot of new insights using that methodology, like the 50x Yamanaka yield that Retro did would be an example of that. But if that's the focus, and the focus is not on generating exponentially more new data than we have, then I guess where does it plateau? Like, do we have an intuition for this?
Yeah, I mean, I'm definitely no, it's a great framing. I think I'm definitely in the camp of I am a realistic but bullish on the future of ML and drug discovery, and mostly bullish because I think there are new and clever ways that we can generate data, and that will actually lead to sort of like the next inflection point. I think I mean, I'll probably be proven wrong potentially, but I guess I'm somewhat confident because I feel like in the last maybe like 2 years we've been somewhat converging on like more models being able to do what other models have been able to do before. I think there's a lot of models now available that are used for structure prediction. You know, protein, DNA, protein, RNA, like co-folding models that can be used as like in silico oracles to do like what I mentioned, like the binder and antibody design. And it's not necessarily that these have been getting exponentially better, but we've just been getting more of them. So the capabilities are being democratized, which is great for the field overall. And I think it will just lead to more people being able to use them and build on top of these platforms. But I think sort of like the next sort of like inflection point will come from people applying clever data generation strategies to expand on what the existing models are able to do. And I guess unfortunately fortunately and unfortunately, I think a lot of these are going to be sort of like privatized because now people understand that data generation is itself a very large mode for companies in this space.
So data generation is the mode. Do you have like a specific example that you could use to illustrate how a company is doing a clever or novel way of data generation in the space?
Okay, this is fun because I guess I'll a bit limited in what I can talk about what we're doing, but I guess to take some of my like favorite examples from literature, we sort of talked about this like paradigm of black box data, which is like an article published by a good friend of mine. And sort of like the idea there is that sort of historically we've been essentially passively accumulating data because humans were interested in them. Like the protein data bank was never designed with sort of like the idea of generating the most diverse folds to be able to train a folding model like AlphaFold. It was generated because people were interested in protein structures, and there was values in being able to, like model a structure to understand how it interacts with other proteins or small molecules. And I guess the interesting nugget there is that the protein data bank always cited to be have cost like $20 billion or some like crazy amount. And this is because there's like 200,000 structures in there, and each of them has cost roughly $100,000. $20 billion is not really in the range of what most companies can spend on data generation. But I think the interesting kicker here is that, OpenFold as a model has shown that you can get to AlphaFold 2 level scale by training on essentially 1% of the data. So if you sort of like take this $100,000 per protein structure at face value, you could actually generate potentially somewhat almost like approximately matched version of AlphaFold. You could have generated it for a fraction of the cost, like hundreds of millions potentially. And maybe if you're clever about the data generation, even less. And that's very much something that companies can do. So like to get back to this black box data paradigm, instead of generating data for human consumptions, which is potentially wasteful in retrospective, we're generating it directly with the model training in mind. I think there are, like interesting examples where for example, if you care about a protein's like stability, like that's not something a value that AlphaFold gives you. I think this is sort of like the side of like AlphaFold has solved like protein structure prediction, but then there's a lot of other properties about the protein that are very relevant for it, where AlphaFold will give you basically no information. One of them is stability. It's like somewhat painstaking to generate protein stability values for a bunch of proteins at like array sort of like one plate scale at a time. So what if you could just pool together hundreds of thousands of protein domains, run like a test for how stable they are, and then just sequence the readout? That might be hard for a human to understand what the output there is, but a model might be able to actually work with this and build like a proxy of how stable proteins are. So what people have done, for example, is like you have like let's say 1,000,000 protein domains, and you express these domains as like, I don't know, like short 30, 40, 50 amino acid mini structures. And you essentially keep a DNA barcode attached to the protein while you're expressing them. So you have this like soup of 1,000,000 proteins, but they're all sort of tagged with a DNA barcode. And now at like multiple different dilutions, you add these proteases, which will chop up a protein if it's not stable, and they will keep it alone if it is stable. So then after you've gone through these like rounds of selection essentially with the protease, you can now sequence everything that's left. And everything that can be sequenced is like massively scalable, right? Because we're really good at sequencing stuff. And now you have a readout of protein stability that doesn't require you to run any individual sort of like test of how to actually measure protein stability. So that's like one example.
So TLDR, black box data, we are able now to generate exponentially more data, exponentially cheaper, as long as we're not worrying whether it's human legible and just feeding it into the AIs to make sense of it on their own.
I think so. Instead of screening like one target at a time, you can express multiple targets with like sort of like CRISPRing out targets in like the liver and delivering multiple viruses at the same time, and then do a single cell readout. And then if your single cell model is essentially able to distinguish a disease phenotype from a healthy phenotype, and let's just say that actually works, then you would be able to screen multiple perturbation at once in like a single animal, for instance. And I think some of these approaches are currently is yet to be proven if they would actually deliver sort of like clinical value. But I think those are examples of like how to creatively just like massively scale up data generation in a way that it would be difficult for a human to understand. We can go into some other examples.
At what stage of proving that it works or having like a good enough indication that it works are we at right now?
That these data generation strategies work?
I mean, you said there is still a gap to be filled, proving that it does something in the clinic. So what is, like in the absence of that, what is the best early indication we have that this is going to be useful and yielding some actual drugs?
I mean, I guess a lot of these companies have sprung up in like the last couple of years, right? And it takes a bit of time to actually get stuff in the clinic. But I guess I think at this point, there's a lot of, sort of like irons in the fire, as they say. And probably in the next couple of years, we'll actually start seeing those clinical trials trickle in.
I mean, maybe to help you out, like, is there anything from these other companies, what they have announced, like what Syra is doing, or some of these big ones, like they have announced a bunch of benchmark results. Like, is any of that like super interesting to you, or I don't know, or do you feel like it's all too early to kind of get excited about?
I mean, I guess like to maybe position sort of my like start with my biases, right? Like we work on aging. I think I'm generally excited about things that I think will actually help us accelerate progress towards longevity therapeutics. And I think the difficulty there is that it's just like not really like one target, one disease, because aging is like multiple diseases. And there's a process that we don't yet fully understand. So I think I'm more excited about approaches that actually like help us model the biology behind what's happening, rather than necessarily knowing an individual target and designing a specific binder or antibody against it. But I feel like we've been making most of the progress on the latter set of problems rather than the former. Of like, there's a very complicated disease process. We can only really observe the healthy and the disease state, and like very little of the progression between the two. How do we actually intervene in this process?
So have we made more progress on that side of things because it's easier to model it, or is it because there's a higher commercial incentive to target cleanly a disease, which leads, I guess, more directly to a drug rather than understand the underlying biology, which is probably a few steps removed from generating revenues and having something in the clinic?
It's definitely true that we have been able to make like a lot of progress on this because it's heavily incentivized. At the same time, I think it's just where the data is. Like I think people, like I'm so as I alluded to before, people have been making heavy use of the protein data bank and the protein sequences, and I think we've been seeing the most progress there in the like being able to start to model cells in a useful way, to be able to do like target prediction, or like, hey, if I overexpress this gene, what happens to my cell? Is it able to go from a disease state to a healthy state? There's a ton of single cell data out there. There's a ton of spatial data out there. I think right now it's just like a lot of progress is driven by like where the public data sets are going.
So maybe just to step back here and to paint kind of the ladder of how we're using AI to model things at different scales. So correct me if I'm wrong, but the way I understand it, like one way to slice it horizontally is that we have these protein models. So AlphaFold is an example, and this is probably the most crowded or like the most exciting space as well, because as you said, we have so much data and we're trying to model how proteins interact. And then one step higher, then we have this, what we're calling virtual cells now. So we're trying to model how the cell actually interacts with different things. And then we could be climbing this ladder few more ranks up, and we could start modeling virtual tissues or virtual organs or whole bodies, which probably, I'm assuming, each step increases complexity and gets harder to do.
Yeah, I also, just to add to what you said, I think there we, I guess like an even like one scale up of the protein models is like DNA models, right? Like we have all of the protein data comes from like DNA data. We don't actually currently sequence any proteins, and we have like assembled a ton of mammalian as well as like fungi and bacterial genomes. And I think these models are also very exciting, although probably the use case there is not like as clear yet, because most, like I guess like you have small molecules and then you have protein therapeutics, and few therapeutics actually have a DNA element in them. I guess RNA is like a bit of a growing therapeutic. I think there's been a ton of progress in the DNA modeling world. It just hasn't really found the use case yet where you have a generative use case and your model just spits out the therapeutic. And I guess like on the on like the layers above cells and tissues, I think what we have done is sort of like use organoids or even like cellular models in vitro to predict toxicity, for example, in vivo. There's companies that are focused on that, on sort of like direct and I think then people are looking at like clinical trial data, and like, can we directly predict sort of like from an input molecule and some patient characteristics, potentially like patient genomes, can we directly predict clinical trial readouts? I think this is sort of like the next scale, and I think the further we go like up the ladder, of course, I think the less data there is, and I think also the slower the feedback loops are.
Okay, so we have got proteins, we have got DNA models, we have got virtual cells, and then above and above and above. And yeah, the data gets sparse, more sparse the higher we climb. So are we, like, what's your intuition here? Is the correct approach to be solving the lowest possible rank one at a time, and does that unlock a higher rank, or do we have to, or are they kind of mostly independent, and do we have to be collecting data on each independently and solving them simultaneously?
I mean, I think it's reasonable for people to go where there's like fast feedback loops and immediate applications. As you sort of mentioned at the beginning, right? Like currently we don't yet have a sort of like AI generated approved drug. That seems like a reasonable milestone to pursue for the field as a starting point. And I also think it's very reasonable to sort of like max out the lower layers, because that will just happen the fastest and then expand from there compared to but I do think the most interesting applications are going to come from people who find clever ways to collect data on the like later layers.
Okay, so we have discussed the protein layer in some depth, and then the cell is one of the next layers up. I've done some digging into things you've previously said, and you said that 95 to 98% of single cell methods right now are BS. I'm paraphrasing. uh, and there's a 2025 Nature Methods paper that backs you. I think it concluded that foundation models couldn't beat a simple linear baseline at predicting perturbations here. So yeah, can you help us unpack this? Is the current state of virtual cells an emperor with no clothes, or are we just too early, and where are we at?
I must have made a great move when I wrote that. I think look, I think it's maybe worth unpacking of like what is the, I guess, what is the goal of a virtual cell, and I also just I guess generally I sort of like dislike this term virtual cell. It's just okay, fine. The field arrived at this like branding term. I'll take it. But I think it kind of like confuses like what we're actually trying to do, right? I think ideally what we want to be able to do is say we are, like okay, like I'm as I mentioned, I'm interested in aging. A lot of aging happens cell intrinsically. Some aging happens in an extracellular environment, or just generally in like the tissue space, but a lot of aging happens on an intracellular level. So if you actually were able to model the behavior of cells, how it responds to drugs, how what would happen to the cell state if you overexpress certain genes or if you knock down other ones, that could actually help a lot with potentially just like screening targets for different age-related diseases or even aging itself. So then the question is like, what data do we actually need to be able to model this? Or even like other things about cells. Like if we're interested to deliver therapies inside of cells, what does this, like for example, antibody drug conjugates? There's like an antibody that targets a receptor on the cell surface and is being internalized. If we were able to model how that happens, that would be very interesting. So what data do we actually need to be able to model this? And so far, the field has sort of just resorted to what data do we have a lot of, and that's single cell RNA-seq data. So, like, can we learn is then the goal to learn a representation of this data and fine tune it, be able to predict whether overexpressing or knocking down a gene will actually lead to the desired therapeutic effect? And so far, I would say, as you've alluded to, we just haven't been able to do that, even though we scaled the data and we scaled the models, and yet very simple baselines still outperform these models, which is just like not true for DNA or protein models. Simple approaches for DNA and protein modeling have not gotten us better antibody binders or have not gotten us like the ability to generate whole new proteins.
So what would be a better, or what would be your dream data set to train on if we had zero constraints regarding the data that we actually have?
I just don't think the field knows right now. Potentially it's single cell proteomics. Potentially we need to be able to model cells across time. Potentially mammalian cells, and also I guess like we're not usually working with primary cells for these models. A lot of them are trained on like dissociated primary cells from tissues, and then sort of fine tuned on like cancer cell lines or immortalized cell lines, which are phenotypically quite different. Like maybe we just need to scale down the complexity and go to simpler organisms to figure out like what data do we actually need to predict like an E. coli or a yeast. And then once we figure that out, we go scale back up to actually be able to do human cells.
So it sounds like the best approach then would be to just continue tossing many different irons into the fire. And that's what I guess all of these different companies are doing in their own ways. And we probably don't know which one is going to yield some really useful models at the end, but that's the only way to find out.
I mean, there's like a I think the problem with the cellular modeling is that with, I guess, protein structure and protein sequences, basically what you see is what you get, right? The data you collect is the data you want to model. Whereas for a cell, there isn't one molecule that is like really able to define all of what it does. You could collect cellular data in many ways. You could image the cell. You could look at the proteome. You could look at what it secretes and what it uptakes from the environment. You could look at the just say the DNA of the cell. I mean, at the end of the day, like the DNA like determines a lot of like what is expressed and in what context. Um, and like the modifications on top of it, of course.
If I understood it correctly, and correct me if I'm wrong, you were saying that, okay, we can create a DNA only model or a proteomics only model, which will each model one specific aspect of the cell based on one specific data set. But then you indicated, okay, cell is a complex system and all of these things interact with one another. So perhaps modeling just DNA from DNA data on its own is not really the way to go anyways. Is that kind of a fair summary of where you were headed?
I think so. I think we currently haven't yet. I guess the assumption. Okay. Maybe I'm back on this train. I think the assumption behind some of the existing models is that a cell is effectively a bag of RNA, and the RNA of a cell is a compressed representation of its state, essentially. So if we model essentially the state of the cell via RNA, we're able to sort of compress all the other functions that are happening and still be able to make useful predictions. And I think so the success of the virtual cell initiatives will depend on to what degree that's true. And I think it's probably more true for some processes than others. Like I do think for toxicity prediction, this seems like potentially a useful proxy to just look at the RNA state of a cell and to stress them with certain small molecules and then be able to read out the perturbation response. And for other processes, like if you're interested, for example, like what receptor a cell expresses or what it secretes, I think it might be more difficult because I think there a lot of the like protein activity and abundance is like quite decoupled from the RNA state.
So cautiously bullish on what RNA modeling can do, but with some important caveats about the limitations.
Yeah.
This podcast is brought to you by Superhuman Fund. If you're building on the frontier of human enhancement, we want to hear from you. Link in bio. We're looking to back the first trillion-dollar company in humanity 2.0. Okay, so we have discussed protein models, we have discussed virtual cells, which are different ranks of the ladder, and we have discussed we've touched on some of the limitations or bottlenecks towards drug development, even if we get these individual layers right. And I would love to double click on some of these. So you already mentioned we're still bottlenecked by clinical trials in order to yield drugs. Like, is that the main remaining bottleneck? And do we have an indication of whether AI helps us to unfuck that as well?
I like that framing. It's even I think there's a lot to do even if we take the things that are already working well. Like take an antibody. Say you now have an oracle which tells you any target, you will immediately get a binder that is able to inhibit and block that target like an antibody would do. There's still the question of you need to actually be able to turn this antibody into a therapy. Like you need to express it. The if it expresses very poorly, or if most of it from when it's expressed just like turns out to be an aggregated gunk, you're not going to be able to translate it very well. You're not going to be able to produce it very well at like large scale. So I think there's like the what people call like developability aspect um, of like translating a known binder into an actual therapeutic molecule. So like the expression aggregation question is one of them. Like another one is, you know, interestingly, um, a humanized antibody would still very often cause immunogenicity because our bodies just like, are kind of evolved to not take up foreign stuff, and that's a good thing, that's a great thing. It also makes therapeutic delivery of, even the humanized antibody still very difficult. And a lot of that data exists locked up in pharma companies. And from the public domain, you would be able to, work with sort of like an aggregated version of that data, but you don't have the full picture. So this is, I guess, like another example of like, if you actually run these trials, if you actually generate that data, you will be able to make those predictions. But this is sort of like the hard part, right? You have to like actually launch like probably hundreds of antibodies into the clinic and collect the right kind of data from patients. Like what are some proxy measurements there that actually have any predictive validity for human immunogenicity? And a lot of the in vitro and like animal assays don't seem to have that great predictive validity. So then we go back to the question of like, what's actually a clever way to generate that data that would have any prediction ability?
So the holy grail of AI drug discovery here, if I understand it correctly, is basically to set up this autonomous scientist loop that generates data, validates it in an actual wet lab as autonomously as possible, and then repeats the process and accelerates the entire loop as much as possible, uh, which would really help us to get through all the stages faster, correct?
I think if we can I'm not even sure if like the sort of like step by step automation of all of the steps is necessarily. I mean, this is like the most sort of like easy to envision path. I do think probably the biggest alpha lies in the domains where someone is able to take sort of like a three-step automation process and turn it into like one single pooled screening system. Like for example, being able to directly screen in like animal models of interest, um, or having sort of like organoids that are actually close to the in vivo phenotype that we care about, and they have proven predictive validity, and we can screen in those instead. Um, I think that would probably be I'm sort of like more interested in those approaches, although it's a bit hand wavy to say that, than sort of like uh, automating every single step of the process.
Is that kind of the non-consensus view in the space?
That automation is hard.
That's we shouldn't be trying to automate the full loop, but just intelligently focus on, I guess, the most impactful bottlenecks.
I think there's sort of one world that I think potentially it doesn't yet exist, but there are signs. And I think if we sort of like further go down this path and actually are able to execute on it as a field, I think everyone will end up being better off. And that's sort of like the I would call it like the like AWS serialization of biotech, where like I always make this like when Airbnb started, right? Like you don't have to build your Google Maps from scratch, and you don't have to like buy all the servers from scratch. You can rent it from AWS, and you can use the Google Maps API to start building the product that actually sort of like differentiates you and that customers will interact with. And in biotech, I feel like oftentimes every company like builds the AWS from scratch, and it builds the Google Maps API from scratch. And I think that's holding back a lot of progress. And we just don't have good APIs right now where someone I mean the APIs are essentially CROs, but I guess the CROs of like are of mixed qualities, and I think it's like a bit of a jagged frontier. where for certain applications you have a ton of CROs, they're very high quality, they're very reliable, and then for other applications it's basically impossible to outsource something and you have to set it up in house. Um, and I think a lot of these services are like too one-off for there to exist a reliable API, and for others is maybe just like it's kind of a boring business and just nobody has done it before. I mean, maybe plasmid source is like a good example of that, right? Like maybe not a lot of people would have estimated that sequencing DNA plasmids was actually like a viable business, and it turned out to be extremely useful. And then once you've done that, you can expand to, you know, adjacent fields like automating RNA-seq for people. I think we'll see more and more of those companies that will be able to do just like, you know, take your predicted binders and measure the binding affinity. Maybe take your protein and be able to measure its expression, which is like really hard to predict right now. And I think sort of like this API democratization of biotech will probably lead to a bunch of progress because it just like makes individual companies much leaner.
I think another good example is Ginkgo. They have been trying to do this, I think, with mixed success for a while, and they are now very heavily pushing into this fully autonomous lab API scenario. And I know they've announced some very interesting stuff with OpenAI. They've been working on that front as well. But I guess to step back, what's your sense? How far are we from this AWS for bio scenario? Are we almost there, or is it? Are we still quite
Oh
far?
no, we're not almost there. Yeah, I think we're quite far from it. Drug development is painful, and
Um,
yeah, I think it's not yeah, I think a lot of it is not necessarily a money or a people problem. I think infrastructure is just like not quite there yet.
and there's a technical bottleneck mostly, or like what is bottlenecking us from having the AWS Cloud Labs infrastructure in place? Because there's a separate bottleneck from having data to generate models. There's a different layer of the stack, right? So what's the bottleneck on the on the physical lab automation side?
I think automation works when you have well-defined use cases that you're going to end up running over and over again. And I think a lot of the automation space has obviously like moved to the high value applications, which is like the clinical side, right? Like there you need something that's predictable, reliable, and it will run over and over and over again. I think so far there haven't been good examples of companies being able to go to the discovery workflow, which is generally like much more variable. You don't generally have an assay that you're going to run over and over and over again. And if you do, you can probably just set it up in house and it will be like the mode of your company. But like most of the discovery process is like running 100 assays every now and then. And those assays have like some overlap across companies. But I guess so far the challenge for the cloud labs has been to find the sort of like valuable overlap that people care enough about and where you can also like actually extract some value and like generate value and capture some of that value. And that's sort of like my model of why there hasn't yet been any economies of scale on the automation side of things.
Okay, so we outlined the bull case where we want to go to. We've outlined some of the bottlenecks. So one being in the bottom AI lab automation, physical automation layer. Then one of the other bottlenecks is on the data generation layer, which helps us train models and understand how things behave. And even if we solve these two layers, then what are still the remaining bottlenecks? Is it then about delivery, making sure that we can deliver drugs into the right places? Is that just part of the same AI modeling problem, which will solve this too?
I think there's definitely like a spectrum between although I don't think it's a huge one, right? I think there's a spectrum between something that is actually an engineering friendly like approach where, you know, like antibodies, we can engineer them and they're broadly useful. So I think we're sort of like, yeah, like the molecules that we're able to I guess like engineer have limitations because like antibodies just won't go everywhere in your body. They're quite bulky. they're going to have limited like tissue penetration of certain tissues, which is fine if they act in the blood. It's less fine if you want to actually get them into specific tissues and potentially cell types, or even across like barriers like the blood-brain barrier. There's like limited examples of antibodies that are able to cross it. And then the sort of like like small molecules that are able to go everywhere, and you can make them extremely specific and you can package them as a pill. We're just not that good at engineering them yet. So I think potentially like closing that gap will be my sort of saying is that in the limit everything will be a small molecule, but your small molecule is probably not going to be the first step of the process because it's just harder to engineer.
Okay, so it sounds like that's a pretty good map of all the fronts we have to tackle, and I guess we're tackling them simultaneously to arrive at this, autonomous scientist loop utopia that's really going to move the needle on the translatability of drugs. I think that's the interesting stat that I was looking at the other day. So drug approval odds, they have stayed flat at about 8 to 10% straight through the genomics, CRISPR, machine learning revolution. So it seems like these technologies have been useful to help us make sure that we we don't get stalled not discovering any new drugs, but it has not helped us break out of this plateau to the upside. And what we are building, the bull case scenario, is that that's really going to move, and the 8% change to 20% and 30% and beyond, right?
I think there's this like spring, which like over time we've been able to layer in more and more and more technologies on top of it, and at some point that spring surely has to just snap, right, and just like unleash the um, power of all of these technologies that we've been accumulating, and maybe I mean also there's always a bias here, right? Because like if like even if that spring pops at some point, it will probably take you 5 years to figure out that it has popped. Um, but I mean, I feel like now is a good time if there ever has been one where we may actually be in this scenario.
What would be the, I guess, the bull case for why now this scenario? Like why are we at the inflection point now?
I mean, I think we're I mean a lot of different okay, like at least two I think one of which is that I think the yeah, I mean we're actually applying large scale models to the domain of biology that humans just like are really bad at understanding, and I think it's like inevitable that AI will eat up more and more of the like molecule uh generation and engineering process because humans just don't really understand a lot of what's going on there. So I think that part, and we have some like clear wins there where models are actually able to do things faster, cheaper, at higher success rates than what we were able to do before. Um, I sort of like mentioned this like wow moment in the beginning for like our transcription factor engineering project, where it had taken the previous academic effort about 10 years to arrive at a sort of like certain mutations to make those factors more efficient, whereas for us using this protein engineering model it was like a couple of months where we essentially like generated the sequences and then tested some of them in the lab and got those results. So that was really like a moment for me where I was like, okay, like we are actually starting to compress timelines now. And then I think the second one that's you know I think overly cited, but I think the GLP-1s are an interesting shift in how we sort of like do drug discovery, because it's a shift from going from, I guess, like concentrated spaces like a huge focus on oncology and like some rare diseases to also look at the like chronic diseases and how to like prevent diseases in the first place, which is something that I'm particularly excited about, and also why I started working on aging in the first place. I think a lot of diseases are best treated by just preventing them from happening in the first place. um, and the GLP-1s are like there's, I guess, a single receptor that you're targeting, and some of the newer versions target multiple receptors. But the mechanism of action is very much you're not just doing one target inhibited, but really there's like a plenitude of mechanisms. And I think the field is waking up to even though those molecules are more complicated and the mechanism of action is less understood, this is really what's gonna get like the large effect size that are required to really like treat and cure many diseases, and as well as aging.
would you dare to put a year on your estimate when the first AI discovered drug actually gets approved?
Approved?
Yeah,
I mean, I think there's a high probability that within the next year, it will enter the clinic, and I guess based on the clinical path that will be chosen, I guess like five years, 5 to 10 years.
5 years from there. Okay.
I mean, to be
Um,
fair, there's one I think going into phase 3 right now, so maybe it will happen sooner than that. But
cool. And I guess one other thing I think you mentioned briefly, but we haven't really dug into, uh, was the so-called anti-scaling law. So there's this theory that actually biology behaves in an opposite way to how conventional LLM scaling works, because we are starting by addressing the most convenient, the most well understood targets that we already have. Uh, Arguably, Yamanaka, that Retro worked on, would be an example of such a well known target. So we'll be able to make really good progress by optimizing molecules to hit those targets initially. But then, as we are running out of ideal targets, it actually gets harder, not easier. I know you have shared that article before, so what are your thoughts on this?
oh, you mean we're I mean, oh, you mean where essentially like there's few targets and those targets will get very crowded.
Yes.
I mean that's kind of already the case, right? I mean, there's like if you look at the like hundreds of molecules against PD-1, EGFR, now GLP-1, so and then you look at some potential like targets in aging and age-related diseases, and there are zero FDA-approved drugs and zero in the clinic. I think that that sort of like crowding is already happening, and I think it's mostly a market mechanism, right? Like taking a risk on molecule, indication, and target. I think most pharma companies want to minimize all three of them. However, back to the sort of GLP-1 story, I guess that was considered a risky target, a risky indication, and sort of like a new class of molecules, new-ish, I guess. And it ended up leading to what will be the biggest drug of all time. So I think the sort of like thinking in the field will switch to instead of going from more target crowding to more approaches that like actually take more risk in the discovery pipeline because the rewards will end up being bigger.
But you are saying the fact that it hasn't happened yet is a function of economic incentives and human decision makers not going after those targets. It's not really reflective of the limitations of AI, or I guess it's not really addressed by AI, but by human incentives.
Yes.
Okay, okay, so that's one side of the equation. Human incentives have to point us there, but then once that happens, that would still be a fairly different capability to start training the AI models on, which is new target discovery, versus what we are currently optimizing them for, which is discovery of proteins or molecules to address existing targets, right?
I think it can cut both ways. I think it can be new target discovery or new molecular engineering that is able to target existing targets that we couldn't tackle, right? I think there was this like breakthrough announcement, I think like last month, from Revolution, if I'm not mistaking this, where you have like a molecular glue which is able to degrade KRAS, which is like one of those targets that people wanted to drug for years, but it's like one of those transcription factors that's very difficult to actually drug. So like now there's a like now essentially someone has broken through that wall and potentially will like by being able to drug targets in a way that we haven't been able to do before. I think it will open up a whole range of new targets, because it's not only that for some diseases we just don't know what the target is. Maybe like Alzheimer's is one of those cases. Maybe there isn't a single target, because it's a much more complex disease than that. And then there's the examples where we know the target, like P53 for cancers. Like we know if we drug P53, we will probably have a large impact on a lot of cancers. We just don't know how to drug it. And I think AI can help with that part.
So AI can help and is starting to help us drugging hard to drug targets. But if I understood it correctly, we are not really optimizing our AI models currently towards discovery of entirely new targets. Is that correct?
I think that depends on the company, right?
Okay. What would be an example of a company that's doing that, that's using AI to discover entirely new targets?
I hope we'll be able to share something about that soon.
Okay. that's that's a good answer. That's yeah. I will stay tuned for that. Um, okay. Um, I think we have circled a lot of really interesting stuff there. I think one lid that we haven't opened, and you have hinted at that, is basically the distinction between AI being used to make sense of large data sets to discover new stuff, and AI being used as this co-scientist to help a human make sense of existing literature and make all of the existing processes faster. So can you help me maybe parse the distinction and share your thoughts on this?
I mean, if you, I guess, for the ones who haven't heard, large language models have become quite powerful, and I think there's a, I mean, I guess I can speak for sort of like my personal workflows and sort of like the lab that I'm leading. Codex slash like Claude Code slash the chat versions thereof have, I think, just taken over a large part of the hypothesis generation, protocol generation, taking raw data and just being able to analyze it essentially instantly. And it just really feels like we're just so much more empowered, where like maybe previously someone would have like run like literature syntheses and they would take in like half a day or something, and now you basically go off and like launch your 10 agents and you get a cup of coffee and you come back and you have access to all the information, um, to the point where I feel like it's so powerful to the point where the models are sometimes their own worst enemy. Where sometimes it's actually like, I think when you're at the cutting edge of research, it's probably like a better idea to do what the model says is not possible, because oftentimes the models say something isn't possible just because it couldn't find anything about it. It hadn't been done before. So then sometimes the model's recommendation is an anti-pattern, but to me it's like you kind of break this by actually giving the model the ability to not tell you like here's what you need to do, but rather like here's some hypotheses to test. And then ideally you can close the loop between a model like saying something, and framing its uncertainty around that, and then also being able to answer it directly, instead of relying on the models to like just like zero shot the answers and then print a molecule and try it. But yeah, I mean like the and not to like, I think there was a time where like bioinformatics, computational biology was, I think, like um, I mean, teams of people in companies would focus on it, and I feel like now we're getting to the point where individual scientists with effectively no computational experience are able to access, maybe like 80 to 90% of these workflows, to the point where specialists are still helpful, and it's still very helpful to have like bioinformatician and computational biologists like set up and command these pipelines. But I feel like we've just like been able to democratize this capability to many more people, which I think is extremely exciting, because it sort of like removes the like a scientist now is just able to actually follow the scientific process mostly, instead of being bottlenecked by like not having the tools to be able to plot something the way they want.
That's very interesting. Is that actually maybe one of the most underrated outcomes of AI in biology? That, as you said, it just democratizes access and multiplies the number of human scientists that can put eyeballs and hands onto some of these hard
I think
problems
honestly,
because
yes.
they can.
100%. And it's like, I mean, like, where do you sort of like put the like maybe individually it doesn't necessarily lead to these like, although sometimes it can, but like imagine it doesn't necessarily lead to these like 10x, like 50x improvements for like all domains. But I would say with some certainty that, I mean, the people who have been just like diving in and using those tools, let's just say like all of them are just like 10, 20% more efficient. And, given that the frontier is somewhat jagged, in some domains you're like 30, 40, 50% more efficient. And then like basically apply that to all the scientists in academia and industry. like those are large improvements in productivity across the whole field, rather than like having a company that maybe like publishes a model that is able to do well in like one particular domain. Here's something that is actually able to effectively help everyone, because that's what scientists do all day, like analyze data, um, generate protocols, like parse through papers to see what has been done before, to be able to then triangulate it and like come up with a protocol. And maybe the protocol now has a 50% chance of like working the first time you try it. That's already like weeks worth of work, because like protocol troubleshooting is a huge time sink for like setting up and testing new approaches. If you capture your data in the right way, and like have the model access that, now it can find blind spots for you, because oftentimes science is messy. And the good thing is the models are really good to like deal with this mess. So potentially it will find blind spots for you, like weird papers from like 1990 that has already like done what you're trying to do, and now you can try this like reagent that you hadn't even considered. um I think this is probably the thing that's like not talked about enough, that's actually the like the less fancy thing. Whereas like it's not like a fancy model announcements, but it's more sort of thing that just makes every scientist like gives them more ability to just focus on the science.
It's a very, yeah, that's a very good way to put it. And so now when I step back and look at uh, what we discussed, my mental model is something like, okay, if we double click on AI in biology, what it really is, it's a bunch of parallel currents, amongst them being AI co-scientists that democratizes access to being able to make sense of data and to access a large number of obscure papers at the same time. Another current is these protein models and cell models, and yeah, all of this across all the different layers of biology we have. Another current is autonomous or semi-autonomous labs that run experiments faster, and maybe several more currents that we have touched on, and they are all kind of interacting and weaving together. And the bull case is that the combination of all these will start to bite at the 8 to 10% drug approval rates and inflect it to the upside, hopefully exponentially, and that's what we are building towards, right?
I am definitely like in the I guess as an AI person coming into biology, um having been burned at the bench, sort of like realizing how, so like the initial ideas you had going in the field, what the actual bottlenecks are, to then facing reality and like, like trying to do establish like new science, and like feeling the actual pain of like how hard it is and where the difficulties is. I think it's like inevitable that eventually AI will just like overtake and augment in a large part of the discovery process. And I think quite excitingly, we're probably at the start where this is actually happening as a true wave. Whereas like before, people were always talking about like, yeah, okay, there's AI discover drugs, but as you said, like, do they actually work much better in the clinic? Like, are they actually addressing the real bottlenecks? And I feel like the question of whether AI's impact on drug discovery is now like just no longer a question, because it certainly has in many ways, and it just like has started in the past couple of years to like really expand. So I think we're um honestly like just getting started. And I think this trend and wave this time is just like not gonna drip off, but just continue and really lead to not just incremental but actually incredible advances in the field. Because again, I think humans are just not we're not like very good at understanding biology. I think Demis Hassabis has this thing where it's like physics, like math is the right approach to describe physics, and AI will be the right approach to like capture biology, because it actually is able to deal with the complexity of the systems that we're working with. And I think that really puts the nail on the head.
It's a good way to put it. And so uh, if I'm a freshly graduated scientist, or if I'm an AI person wanting to go into biology and make the best dent possible on all this, like what would be the most underappreciated thing to focus on?
Figuring out new ways to collect data to solve like the sort of like bottlenecks that nobody's focusing on right now.
And to flip that, to ask you the spicy question, what is, in your opinion, the most overhyped part of all this right now?
We probably don't need another protein folding model.
Too crowded or solved enough?
It's just like, I mean, it's just like the residual value that can be provided there. It's just like at this point, I think there's a lot of great companies doing great work and a lot of open teams now publishing open work. It's just like I think we're probably already going in the right direction there. I think at this point, if you actually wanted to make a big impact, probably it makes sense to go into spaces that are less crowded.
So be more ambitious and venture outside the box, which I guess is more risky, but that's probably where the next really huge breakthrough will come from.
For sure.
I think that's fair.
Thanks so much for doing this. Thanks
Cool.
for taking the time.
By the way, if you're enjoying this, hit subscribe. We drop a new episode weekly, exploring how AI, biotech, and human enhancement are shaping the future. And if a friend would love this too, send it their way. It really helps us keep going.